ಇದನ್ನು ಪ್ರೆಸ್ ಮಾಡಬೇಕಲ್ವಾ? ಓಕೆ. Good evening all. How are you? Fine. Okay. So online people, good evening. So this class is live, so everybody can watch this. So since this is a live class, so what I will be doing is that, as we know, like paper 1 is completed. So from today we will be starting this paper 2. So before we go for like discussion of genetics, just like in brief I will say like things about this thing, paper 2, right? So as we know, these are the major subjects in paper 2. That is, okay, cell biology, plant genetics, so plant breeding, and then we have plant physiology, horticulture and landscaping, plant production, food production and management system. In fact, this portion, food production and nutrition management, it coincides with GS3. GS3 agriculture portion, which is there in the economy, so it will be almost same, right? So now here, speaking of the trends, what you have to do is that, see, majority of the questions it will come from plant genetics, breeding and horticulture. So combining these three together, out of 400, somewhere you will be getting at least 200. So that is to say 50% of the total paper weightage itself is in these three subjects, correct? So saying that, it doesn't mean that rest of the syllabus is not important. How they will make it important? Because they have question 1 and 5. What they will do is that, so from the rest of the topics, they will pick one question and they will put it in the question 1 and 5, which will be a bit different or else bit difficult, correct? So this is the general trend we are following now. So since we are going to start with this genetics, first I will let you know about the genetics trends first, right? Okay. Okay. So this is the syllabus which we have. So see, I told you in the first session also, in paper 1, syllabus itself is very, very important. If you are really clear about the terminologies they have mentioned in the syllabus, that much is enough. Okay, so even before that, okay, let me clarify certain fears you have, right? Especially in paper 2, like when they come for like mentorship, the first question they will be asking, or else the first doubt they will be having is that about genetics and breeding. So they will be saying, sir, molecular level questions. This is difficult, and for us in UG we didn't have very good faculty, we don't have conceptual clarity, we don't have notes, we don't know how to give examples, don't know how about intro and conclusion, correct? See, among all these fears, I can say somewhere these two I can understand, it is relatively rational one, but rest of the things actually it is, you have made these fears for yourself, right? Why? Because see, if you go through this syllabus, and for most of the questions, like let's say if you take 10 questions, at least 9 questions, UPSC is sticking to the syllabus itself. There might be one question from where they may ask about molecular level questions, but what are the students doing? They are focusing on that one question and they are missing out on the 90%. So this is the biggest mistake they are making. Are you getting? See, at the end of the day, what we want is at least 50% of the total marks. If you are getting 50%, it is more than enough. So that is the trend, right? So that's why for that, again, like 50% is the margin we have, but still we are preparing for 90%. That is good enough. Why do worry about only that question? Are you getting, right? And here also, like speaking of conceptual part, see, for example, they have mentioned keywords like this, heredity, correct? Then what we have is, say, genetic material, that is DNA, RNA, correct? Chromosome, and then linkage and crossing over. See, I am taking just the major ones, heritability, cytoplasmic inheritance and self-incompatibility, correct? And sterility systems. Now my challenge to you is that just go for PYQs, take at least 10-year question papers, and you can find that at least only these topics only is asked. Are you getting? At least for the 90% of the questions it will be focusing on this, right? But what you are doing is that, let's say for example, sir, once there was a question on this gene interrogation, Once there was a question on, let's say for example, interspecific hybridization, and it was asked for 20 marks, how can we solve that? That were exceptions. Are you getting? Believe me, those friends who were of mine and had like PG, PhD in this, genetics and breeding, they still face a problem while writing those answers. Are you getting? At the end of the day, as I said from the first class itself, it is about understanding. Depth of understanding is important. If you understand really what is heredity, and it is having linkage with heritability, right? If you want to understand all these things, first thing is clarity about chromosomes. If you have clarity about chromosomes, then you will be knowing about DNA. Are you getting? Because all these things which I have like noted down here are interlinked. Are you getting? If I want to check whether you have the knowledge of, let's say, genetics, first thing I should be knowing is what is the fundamental thing I should be testing, whether to judge you know about genetics or not. Cell, yes or no? Cell biology. The cell organelle structure, how it functions, where it is, etc., etc. And go back to PYQs, at least one question from cell biology will be there. It might sound very, very fundamental, but that is really, really needed. Clear? So one question from cell biology will be there for sure. Clear? After that, see, if I want to put it, and I have given this in this, workbook also. See, if I want to prioritize this, like from highest to lowest, by the frequency of questions they are asking. So first topic I would be saying is this thing, heritability, sterility, incompatibility. Right? See how beautifully they have framed this syllabus is that they are given three terminologies. One is heritability, sterility, and incompatibility, and they themselves have given this. One is classification of it, second one is application. If you go back to PYQs, again only on these two topics, I can say 80 to 90% of the questions have come. So rarely what they have asked apart from this is that, okay, explain the genetic basis, explain cytological basis, or else, let's say for example, if you take this incompatibility, they might have asked about mechanism, explain how this incompatibility works. Right? What are the different systems of incompatibility we have? But in a way, if you understand the concept, are you getting? In concept only this mechanism is there. Yes or no? They need not mention it exclusively, but they are sticking to the syllabus. Are you getting? At the end of the day, we cannot challenge UPSC saying that, sir, you are given a syllabus and you are going out of the syllabus. Because always we have that luxury. We can say that, okay, this is the applied part of this. So I am been in the syllabus. So it is going to happen. At the end of the day, they will never go out of the context. Clear? So these things you have to keep in mind. So this is the first thing I can say, most important one. After that I can say is this thing, where is that? Linkages. So this is where another question is coming. Then polyploidy, then mutation, then this cell structure function, synthesis, and then cytoplasmic inheritance, then chromosomal structures, and then law of heredity. So this is the order in which they are asking the questions, depending on the frequency of the questions I am saying. Are you getting? Right? So make sure that all this syllabus, or else words, terminologies which is present in the syllabus, it should be there in your brain. Are you getting? Believe me, for most of the answers, today also I am going to prove, because I have done that in paper 1. If you have observed, what I was doing is, I was taking one concept and I was linking to some other concept from some other subject. Are you getting? Here also you can do the same thing. Right? But for that, you should be clear about the terminologies and the intention of the question paper setter. Clear? Yes? So with this we will start this trend analysis of this. With this we will start our genetics discussion. Okay. Plant genetics. Right. So as I was doing in paper 1, first I will let you know how many questions are coming, or else what is the weightage of marks it is coming from over the years, right? So from 2017, 18, 19, 20, 21 and 22. So 17, 70, 60, 70, 65, 35 and 70. See, if I can quantify this, on an average at least somewhere around 55 to 65 marks is definitely going to come from this subject. Are you getting? And in that also, if I want to classify like this, one is fundamentals and second one is application. right? Fundamentals will be more than 60%. See, especially if you go to paper 1, it will be more of fundamentals. Are you getting? Because here there are a lot of concepts where I can frame the questions. Are you getting? If you are not knowing the concept, then it will be difficult. But this application, what I meant is, let's say for example, today also I am going to discuss that question. Let's say DNA. So DNA as a concept, structure, etc. you would have known. So what is the question they have asked? DNA and its role in development of traits, different traits, correct? So this is one type of application. Second one, let's say for example, another question was there, this thing, Aneuploidy. So concept, types, etc. we know. But what they have asked is, they are given the statement saying that Aneuploidies are generally weak, but still it has potential with respect to benefits, correct? So we have to analyze this. Are you getting? So these are the exceptions I am saying. Are you getting? Rather than exceptions, what I can say is that if you are really strong with this 60%, where let's say for example, you know the concept of aneuploidy in detail. In detail is like, not like let's say for example, going to the molecular level, studying the atoms and going subatomic, that is not what I am saying. So if you understand what is the basics of it, that is more than enough. Are you getting? So today I can prove with that concept itself. Clear? But if you are really clear with the fundamentals, so second part is dead easy. Clear? See, why I am taking this much time to emphasize all these things is that it really actually feels bad for me, because when students come and they will decide another optional based on these two subjects only, right? So they are that much fearful because they think that breeding and genetics, if they don't know the agriculture itself. That is really far from any truth, right? So that's why if you have that phobia, forget it. Like today also I am going to prove, like in genetics, just by understanding fundamentals, how you can write the answer. Are you getting? So that's why I taken the tough questions only. Like it is a mix of tough plus easy questions, right? So now with content, how can you present? And without content, let's say just you have the fundamentals, plus still you can make a good answer. How can you do that? That I am going to prove. Clear? Because see, these things which I am saying, it is called as art of answer writing. Art of answer writing. I am saying it as art because it is very, very subjective. Clear? Very subjective, because the way I can frame the answer, it will be entirely different from you, right? So that is the beauty of this examination. Clear? Right? So with this, okay, so I will say this question 1 and 5 trained. So usually in like 1, the question will appear, that is question number 1, that is at least 2 questions will be there. So another trend is that what they will do is that first part, like let's say if at all the question is having 2 parts, so first part will be of genetics, second part either it will be of breeding, or else either it will be of biotech, or either it will be of resistance, right? Could be for pest, could be for disease, something, right? So genetics plus this subject, this is one trend, right? At least 1 question, or else 2 questions of fundamentals in the first question itself. Are you getting? But in question number 5, usually it will be of which subjects? Which subjects? It will be of mainly physiology, some part, mainly plant protection, horticulture and food science. So usually it will be the case, but mostly it will be from this plant protection, horticulture and food science. Clear? First part is most of genetics, breeding and seed science. So this is how the questions are coming. Clear? Right? See, in a way, actually when we are discussing from like let's say first class till to today, what has happened is that I have been telling you like from which part you can expect the question and what is the trend. By this, what will happen is that the fear that you will be having about like question paper, what will be asked, that anxiety will be there. But if you really been through this PYQs, what will be happening? You already know in question number 1, what are the expected things. We cannot say that exactly this question will come, but definitely we know that from agronomy at least 2 questions will be there. So that satisfaction will be there. Yes or no? Are you getting? So this is the advantage of our optional. Clear? Yes? Okay, so you have like any other general phobia about this subject as such? No? No, right? Okay. Even if you have, as I said, see at the end of the day, all that you need is fundamental plus application, right? So that's why focus on fundamentals and it will be good enough. So with this, we will start this session. So first, like you've been acquainted, so for those who are like, for the first time they are watching on YouTube, for them I will say, see, this is my Telegram ID and these are all in caps, capital letters, right? So you can like ask your doubts, etc. here. And another thing is that how class progresses is I will give one question, correct? I will give 2 or 3 minutes, so you can frame your answer or else structure. After that, I will discuss the question, the concept part, and then if at all something is important, I will let you know. I will dictate you few things. Clear? Okay. So then we will take this first question. Question number 1. What is cell cycle? Explain different stages of, see that is not interphase, just mitosis, because we know interphase is a different one. Yes, and this is for 15 marks, so just scratch it down, so that interphase is not there. So it is just stages of mitosis, right? So take 2 minutes and give me the structure. I just now explained Arvind, so how can you prepare for the class? Okay, we will discuss. Okay, So what could be the structure for this answer? Yes? Okay, definition of cell cycle, correct. Then diagram of cell cycle diagram, okay, then okay, okay, stages, fine. Right, so this is the usual structure, like anyone will be having the same structure, right? So here the problem, or let's say challenge, is that, so within this what are the things you are going to fill, right? So that is the challenge, because this is a technical question, so you have to address it technically, and that is the structure we can have. Let's say for example, okay, intro. So what is the definition of cell cycle? So here, as I said in the beginning also, since it is about what and define, right? So better way is to start with the definition itself, yes or no, right? So what is the definition of cell cycle? Or else let me ask in this way, what are the technical words I should be writing in the cell cycle, in the definition part? Yes? Okay, so before cell division, see, think from this perspective, you know cell division, I mean cell cycle. You said that you can write the diagram. In the diagram, what are the things you are going to write? Correct, different stages. So main things which will be happening, first one is synthesis or else duplication of DNA or else genetic material, correct, this is one. Second one is division of cytoplasm, correct, yes or no, cytokinesis, we know, right. After that, final one is, what will happen in general, forget about meiosis, in mitosis it is two identical daughter cells, right. Now utilizing these keywords, can't you make a definition? Yes, cell cycle is a series of events in which the cell organelle or else within the cell, it will be synthesis and duplication of the DNA, which is leading into the division of cytoplasm, and ultimately it will be leading to the formation of two daughter cells. Are you getting? So see, if you remember in this way, you need not by heart the things. Especially the fear in paper 2 is that, sir, there are so many technical definitions, how can I mug up? There is no need to mug up at all, right? So this is how we can remember, right? So this is the definition. Okay, we know, rather than putting the diagram, so I will say like what are the phases of this cell cycle, or else parts of this cell cycle. One is, from where we should start? Interphase or else GO, right? So GO, second one is interphase. In this what we have? G1, yes, and G2. So this is the right order. Third one is mitosis and meiosis, right? So this you can show either in like, let's say, circular manner, flow chart, anything is fine, right? Make it at least like, what is that? Pleasing on the eye, right? So don't make it absurd. And also I have given one of the flow chart in this Telegram group itself. So there you can see. So GO, it is almost like resting phase, correct? But the preparatory work will be like done here, right? So interphase, it is the major one, correct? So in G1, what will be happening? G1, will the synthesis happen here? Will the synthesis happen in G1 phase? Yes or no? Yes or no? See, mainly the synthesis will not happen here, but a marginal content may be produced here, maybe, right? And the preparatory work, that is the enzymes needed, etc., those will be synthesized here, correct? After that, in S phase, this is where replication or else duplication of DNA will happen. So that is why it is called as synthesis phase, correct? Because synthesis will happen here only. In G2 phase, what is happening? G2 phase, again same thing, minimal or else no replication, but it will be helping in preparation for mitosis, next stage, correct? So that will be happening in the G2. And we know in mitosis and meiosis what is happening. So here 2 daughter cells, here it is 4. So it is equational one is what? Mitosis or meiosis? Equational. Mitosis is equational cell division, correct? So meiosis is both equational and reductional cell division, right? So remember that. So these are the basics we have to establish, correct? And this is for 15 marks, so like half of the page definitely will be taken here, right? And since it is just asking about Stages of meiosis. We know how many stages are there? 4, correct? Prophase, metaphase, anaphase and telophase. So 4, think like this, okay. 4 phases are there. Let's say under each, if I write 4 points, so how much pages it might be needing? Half page, correct? Then you have the diagram. Let's say if you are putting, so 3 pages, let's say half page, this much have taken for definition and this diagram, etc., and writing something, right? So here if you are going to give this stages of meiosis, so this is what it is happening, this is what it is happening, and then here what you can do is that at least explain the major things which is happening in the each phases here, right? Because half page is more than enough for you to write about that prophase, metaphase and anaphase, right? It is not meiosis where within one stage there are multiple stages. Here it is pretty simple, correct? So that's why use space wisely. Are you getting? Are you getting? Right? So here obviously, like for example, this prophase, metaphase, anaphase you will be knowing. So rather than explaining on that, so how can you conclude this? Conclusion. So this will be sort of new for you, or else I can say it will be known to you. I will be forcing you to think in a certain way, right? How can we conclude this? So question was on cell cycle plus mitosis. Yes? Yes? How can we conclude this then? Think something, you will get it. No? Shall I give you a hint? Okay, why should we study about cell cycle or else mitosis? Why should we study? What is the use? What is the use? Just for fun we are reading? Or else it is in part of syllabus, that's why you are reading. What is the use of reading this cell cycle and mitosis? See, you know, if I say, then you will be realizing, oh, correct. So, but just think like that. For example, what is mitosis? 2 daughter cells will be produced, or else let's say for example, increase in number is happening. So somewhere, if at all it is increasing at unprecedented levels, it is called as cancer, yes or no? Tumor. Tumor is what? Uncontrolled cell division, correct? And it will be happening at a rapid phase. It is nothing but increased mitosis. So this is one application. Or else let's say for example, proper understanding of cell cycle and mitosis helps in knowing about, you know, this phenomenon called as cell signaling. Cell signaling. So for example, can you give any chemical or else like this thing, what is that? Agents which are involved in this cell signaling? No? Have you heard the cyclins? Cyclins? No? So at least you should be knowing these things. Because in cell cycle, these are the fundamentals I'm saying, not like innermost things, right? So cyclins, so let's say the same proper understanding about cell cycle and mitosis helps in understanding about cell signaling like cyclins, which finally or ultimately helps in oncology development, correct? We can relate it to cancer and cancer therapy. Are you getting? So this is the fundamentals, right? So that's why, see, when especially I can also say during when we were like during our UG, we also thought of the same thing, because see, genetics and breeding, it is such lively subject. If you understand it, you will enjoy it to a maximum extent. If not, you will be totally out of the context, right? So for us also, unfortunately same thing was happening, right? So now let's say for example, when I say certain concept, right, if I say why you should be reading this, what is the practical utility of this, then won't you be excited to learn? See, that was missing. Are you getting? So here, this is the practical application. If you give this in the conclusion, won't that be adding as a value? For sure, 0.5 I can give you extra, because what this is highlighting is that it is not just about application, it is about conveying to the evaluator that you know the concept. Are you getting? Only and only when you understand what exactly is cell division and what exactly is mitosis, then only you can relate to this sort of like concepts. Clear? Yes? So this is what I said in the beginning. Even for the like technical sort of things, you can definitely bring introduction and conclusion. Are you getting now? Yes? Examples of? Okay. Would it be wise to use that? See, what you said is correct, but see, for example, if at all it is on Drosophila, though technically it belongs to insect, but still you can use that, right? That is one. And let's say if at all the concept is such that it is mostly explored in the like human area or else like animal health area, definitely you can utilize that, no problem. See here we are studying this as a concept, right? Unless and until it is exclusively used or else most commonly used in agriculture, we cannot force that concept to be used in agriculture. There will be practical limitation. Or else what you can do is that you can give that itself as a like this thing, what is that conclusion, saying that most of the work has been done in the animal genetics, especially on this part. So there is lacuna in this in plant kingdom, so it has to be fulfilled. In that way also you can bring. Or else let's say for example there will be practical limitations. Why that particular concept cannot be explored in genetics, I mean plant genetics. Focus on that limitation. Why exactly it is not happening? That would be better, right? So rather than entirely eliminating the option of writing those examples, better will be this thing. Clear? Right? So then we'll take question second. Okay. This is good question. DNA is a genetic material. How does DNA contribute to development of various traits? This is for 10 marks, so 2 pages will be there. So take 2 minutes, then give me the structure. Okay, is it done? We'll discuss. Okay, DNA is a genetic material. How does DNA contribute to development of various traits? See, the gravity of this question, it is very, very important, because let's say, see, recently there was this documentary also available, and we also know that James Webb telescope was launched, correct? Have you heard? Yes? Have you seen the images of that? What is it has sent back? Just go through that, because it is very, very beautiful, and in a way like it is achievement for the mankind, right? And this is nothing but an amalgamation or else accumulation of, that's for what we had accumulated about space travel, that's for what we had known about space as or else universe as a whole. Are you getting? Yes, right? So now to evolve to this, before this we had, which was the telescope? Right? Hubble telescope, correct? So see here the problem was, it was in like orbit which is like very near to Earth, right? But this is going into deep space, correct? So before that this gave us certain idea, and even before that, let's say for example, if you have watched like Mexico or as let's say this in Antarctica, we have this big telescopes on this Earth also, right? So the knowledge which we gain from this also has contributed to this, correct? Let's not forget about the microanalysis. We had microscope for the beginning. We know Robert Hooke, he saw it, cells, yes? So then we got to know that we can see the world in microscopic level also, and we can see the things which is not at all visible for us, which is millions of miles or millions or light years away from us. Are you getting? So what I am trying to highlight here is that, see, this is the fundamental unit from which we are all made up of. In fact, this is the fundamental unit which the entire universe is also made up of, because at the end of the day, if we divide all these things, it will be going to this chemicals only. Yes or no? Carbon, hydrogen and oxygen. So this is what basically we are all made up of, and the universe is also made up of, correct? Now what I am trying to highlight is that, see, where is this and where is this James Webb telescope? But all these things are interlinked. The knowledge of one is helpful for knowledge of another, right? So same thing can be applied for our genetics also. How? See, the question is saying that we have DNA, right? We have DNA. And in fact, even before we got to know that, yes, DNA was there, there were so many other theories, correct? And once we got to know that, okay, DNA is there, so it has to be proved as genetic material, which is responsible for carrying of the genes. So this has to be established, and somebody took that burden and they have done it, correct? So now once they have done it, we want to know that, okay, what is the use of this genetic material as such? Are you getting? So one of the uses, development of traits, because what I do, what I behave, it is basically because of my genetic composition. Are you getting? And if I really understand about DNA as such, won't I be solving this question? Are you getting? So this is what exactly this question is demanding, right? So you have to understand the beauty of the question in order to write beautiful answer. But the burden of students is that they will always think that is from the rectification. If you are really biating, you won't really enjoy the science. If you really enjoy the science, believe me, like there is nothing more pleasureful than this. Clear? No. How can we start this answer then? Yes. And forget about those things. For answer, see, when you are writing the answer, it has to be cost to benefit ratio. But that enjoyment should be there when you are preparing. Are you getting? So this is like being Buddha also and being Ashoka also. Because Ashoka enjoyed all the pleasures. Then he became Dhamma. He started to propagate the Dhamma, right? So like when you are preparing, you should be like the pleasure one. That is enjoying like Ashoka. But later onwards, when you are writing the answer, you should be like Buddha. Right? So that is the difficulty, but again you have to achieve that. Clear? Okay. Now, tell me like how can we start this answer? Or else interstructure for the answer. Yes, you got to know about the question, right? What it is demanding, etc. Now tell me the structure. Good, very good. Good. Okay. Good. Then second part. Okay. Okay. So there content might not be there. See, I will tell you, if you have what you said, these two things, it is very beautiful, right? So if you really understand this, not rectify this, you can write this content. How I can prove it, right? So this is what I wanted to highlight in genetics and breeding. So this is really easy question, right? So as she said, what we can do is that, see, even before this, what is that? Hershey and Chase, right? Hershey and Chase. So for those who don't know, so these two are different people, right? So they did this experiments to prove that DNA is the genetic material, right? Now, even before this, you would have heard about this Griffith experiment. Griffith, Avery, and MacLeod. Correct? So these three people individually, they did this experiment and they were convinced to the world that yes, DNA is one sort of thing which is helpful for genes to be traveling, right? So this they had done, but that was not convincing proof to the world, right? So that did not like that had lot of like, let's say ifs and buts, or else like postulates, more of theoretical considerations. But so Hershey and Chase, they gave this undebatable proof, right? What they did, they they took this radioactive phosphorus and radioactive sulfur, right? Okay. So what was the outcome? It is simple. Let's say for example, what is DNA made up of? We know now, right? So what is DNA made up of? Nucleic acids, nucleotides, okay, then nitrogen, phosphorus, correct? So these are the major compositions, correct? If I use this radioactive phosphorus in a organism, if I basically create this mutation using this phosphorus, so that mutation or else that change should be carried to the next generation, yes or no? Because phosphorus is a part of DNA. But if I do the sulfur analysis or a sulfur treatment to the any organism, what will happen? That sulfur thing will not be reflected in the another generation. This proves that because there is phosphorus and DNA is there and protein will be formed, and these proteins are the ones which will be carrying this codes, correct? Basically, because of this we can say that DNA is the genetic material, right? Are you getting? Right? So this is what, like for example, for the introduction, what you can do is that you can start with this thing. I can say like when this Mendel's laws were rediscovered, so like world was fascinated with the genetics. But even before that, people were contributed by discovering the genes, right? And there was debate whether like genes is the genetic material or not, or else DNA is the genetic material or not. So Hershey and Chase provided this theoretical and practical proof which is undebatable. After that, explain this theory in very brief manner, right? Once you have done, you have established the first statement saying that DNA is the genetic material, right? Without doubt, you are establishing the first part, correct? And this is for how many marks? Okay, 10 marks, right? So like this would have taken you like, let's say for example, more little bit more than quarter of the page, right? Now after that, if you want, you can give the properties of DNA, right? Properties of DNA, right? After that, the second part. So second part is how DNA contribute to various traits. Okay. What is the meaning of trait? What is the meaning of trait? Characters, correct? Now, if I say, let's say for example, okay, if I have this diabetes, right, it could be like induced, that is like self-acquired, or else it could be from my parents to me, correct? Or else let's say for example, if I enjoy being alone, so that is also like nothing but genetic expression only, correct? All those things, basically I can say the characteristics are embedded in my genes, correct? All these genes are based on, present on, where are genes present? Fine, it is on basically on chromosomes, correct? And we know the structure of chromosome and what are the mutations, etc. things will be happening here. Based on that, I will be having this, let's say the character modifications. Now, for example, let's say DNA. What is DNA? Basically, it is like a code. I can call it as genetic code, correct? In genetic code, what is happening basically? So in DNA, like nucleotides, what are the major nucleotides we have? Adenine, thymine, guanine, and cytosine, right? So these are the major ones, and if you know like any genetic code, so these are the ones we are utilizing to write. And let's say for example, if I know like DNA composition, and if I alter that, can I be altering this genetic code? If at all genetic code is altering, won't that trait or else character will be altered? Are you getting? Now let's say for example, how DNA contributes to this various trait development, I can say about genetic code, correct? So genetic code is nothing but a sequence of DNA or else nucleotides. So based on the composition of that, it will be leading to different trait formation. Yes, so genetic code is one. And also we know about this thing, what is that? Translation, correct? So translation is the process of protein synthesis, and essentially protein is also what? Made up of, ultimately it is also DNA, right? So genetic code is very important for this also, correct? If you remember, we have this open frame, remember, protein synthesis. So this ribosomes will come, they will sit, they will form this, correct? Remember? So that much should be known to you. So that's why, so protein synthesis, that is also needing to this. Or else, let's say for example, this hereditary diseases, or else genetic diseases, correct? This can also be said, yes, right? And also evolution can be understood. Evolution of characteristics can be understood, yes or no? Let's say for example, food habits, it is also having implication on genetics, correct? If you go to Italy, European nations, basically their diet is wheat based, correct? But if you move to Mexico and all, it is basically maize based. But again, if you move to Asia, it is rice based. So that is evolved based on the genetic composition of us also. Are you getting? Now you got the idea how we can interrelate the things. See, definitely if you go and Google down, you cannot definitely find this content. I can challenge you, right? Because if you want, you can try it down, because this actually what it demands is understanding, right? If you had really understood what is the composition of this DNA and how it was proved to be genetic material, then definitely you can make up this content. If not, believe me, it is not at all possible. Are you getting? Considering that if you have this basics clear, then this question was easy. If not, this question was tough. Based on this, again people will say that I should not choose agri as the optional. Who is the loser? They are the losers, because this is one of the easiest question. Yes or no? Are you getting now? Yes? For this, how can we conclude? Conclusion? How can we conclude this question? We are speaking of DNA. So, where is that? Okay. We are speaking of DNA. We are speaking it as genetic material. We are also saying it for trait development. Now can you think of all these things and make a conclusion? Just now, like in the previous question, I let you know how to think. Now think in the same way, you will get the answer. Okay, one is plant breeding program. In that, can you be specific? Yes? Can't I say like disease resistance, pest resistance? Definitely it can be done, right? So these are also what? Genes only. Yes or no? It will be imparting immunity to me, right? So this can be said, correct? Right? Anything else? Good, very good. Human Genome Project. So this also we can interlink here, correct? And anything else? Any other practical utility? Yeah, like, see for example, when this Cas9, very good, very good. So that is where I was thinking. Let's say for example CRISPR-9. So when this technology came, basically what we were thinking is we can edit the genes. If you are editing the genes, what is happening? We can eliminate this hereditary diseases. Yes or no? Right? So that can be said in the answer itself. Yes or no? Because we can say that the hereditary diseases can be removed or else eliminated by Human Genome Project, where we are analyzing the analyzing the genome of humans. Same thing can be said for plants also. Yes or no? Because see, we know Drosophila, it has been mapped properly. We know the genome of Drosophila. But same thing cannot be said for other crops. So once we have this genome analysis of one plant, so based on the DNA modification, definitely we can bring out the goods. Or else, if you want, you can also interlink like this. The knowledge of DNA combined with applications of biotechnology, evergreen revolution, or else let's say for example, elimination of hunger, or else elimination of poverty can be achieved. Yes or no? Right? One is technical part, another one is GS part. Again we can interlink them. Are you getting? Right? And there is no hard rule that you cannot interlink like this. If at all it is adding value to the answer and bringing justification to the demand of the question, definitely can do so. Clear? Yes? Yes, clear? Enjoying? Feeling sleepy? Okay, third question. Okay, else I will just dictate few things here because there is the DNA thing, right? I will tell you how this DNA will influence the trait development. Just you can take it down. First one, I'll put heading as how DNA influences development of traits. How DNA influences development of traits. First one, genetic code. Genetic code. DNA is made up of 4 nucleotide bases. 4 nucleotide bases. ATGC, adenine, thymine, guanine, and cytosine. Full stop. The sequence of these bases forms genetic code. Forms genetic code, which are essential for many cellular processes. Second point, gene expression. Under gene expression, for any gene to be expressed, 2 processes. One is transcription and translation. Right? So take down under gene expression. One is transcription. RNA polymerase reads DNA sequence of a gene. And synthesizes a complementary RNA strand. Next, translation. With the help of mRNA and tRNA, protein synthesis will happen. Next point, interaction of proteins. Different proteins play a role in controlling growth. Providing structure. Catalyzing biochemical reactions. And regulating various physiological processes. Next one, complex traits. The inheritance of polygenic traits have complex interactions between different protein molecules. Clear? Yeah, next question. Third question. Describe loss of heredity. Various loss of heredity. Explain law of independent assortment. Independent assortment with example. So this is for 15 marks, right? Yeah, this is for 15 marks. So 3 pages will be there. So give me the structure. Is it done? We'll discuss. Describe various laws of heredity, explain law of independent assortment with examples. So this is like one of the most repetitive questions in genetics, right? And though it will be asked for like 100 times, 101st time also it will be asked. Because again, this is like bedrock foundation for this genetics as such, right? So that's why this concept as such is important. Clear? Okay. Now, what is the structure for this answer? Yes. So this is for 15 marks, so 3 pages will be there. So keeping that in mind, can you give this structure? Yes, see, I can know, like it is pretty straightforward questions. We know that 3 laws of heredity is there. We have to explain that, and then explanation of this independent assortment, right? So that will be the structure. You may say, sir, we will start with something intro, then this 3 laws, and then those independent assortment, right? So that is correct. So again here, like what else the better things you can do in the answer. that I will be telling. So how can we start this answer then? Introduction. Works of Mendel, okay. One is Mendel works, okay, fine. Then anything else? Any other way? No? Only one way. Can we define heredity? Yes? Can we define heredity? Then we can link to this. That will be much better. Correct? So now what is heredity then? What is heredity? Yeah, what is heredity? Passing of genes from one to another. Can I put it in matured way? Can say that this thing heredity is genetic heritage one can derive from biological parents, right? It makes more sense, correct? So we can say that heredity is the genetic heritage which one takes or else which inherits from biological parents, and it was proven by the efforts of Gregor Mendel via experiments on the pea plant. So that would be better. After the experimentations, Gregor Mendel has quantified the laws of heredity into 3 parts, correct? That will be developing the context for the answer, correct? What are the 3 laws we have? Laws of heredity. What are those? Come on, these things should be on your fingertips. First one is assortment is 1, second segregation, third dominance. In fact, we can put the first law is segregation. Segregation, then assortment, then dominance. Correct? What does this segregation say? See here, I assume that you know these fundamentals. For example, what is gene, what is allele, where is chromosome, all those basic things you should be knowing. So that's why cell biology is the foundation for genetics. Clear? Okay, so what is segregation? Or else what does this law of segregation say? Everybody has forgotten. See, for example, each trait or else characteristic, it will be having how many alleles? 2 alleles will be there, correct? So now what is this segregation is saying is that, so each allele, like one allele from each parent, it will be segregated and there will be no mixing. Are you getting? Let's say for example, this this is one let's say trait, right? This has 2 alleles, R and R. So same thing could be for this also, correct? Now while this thing fertilization, after fertilization, when the segregation happens, so R will come and this R will come and there is no mixing. It will be independent. So this is what the segregation is saying, right? In fact, the understanding of segregation is important for understanding the assortment. Are you getting? Now what is this assortment is saying? So once we have this this sort of gamete is formed, right? Now once another gamete is also there, when these 2 things mix together, there will be independent assortment. There will be independent assortment. So this is what it is trying to say. And what is this dominance? What is dominance? It is pretty simple, yes or no? When there is, main condition is heterozygous. When there is heterozygous condition, the dominant gene or else dominant allele, it will express and suppress the expression of recessive allele, right? So terminologies are important in these definitions. Are you getting? Yes? Got the concept? Now under this what you have to do is that, because this is 15 markers, at least for 1.5 page you can say that introduction, like heredity and related to like Mendel thing, and then 3 laws. First one is the postulate, that is the definition part, what exactly it is trying to say. After that, just by taking one example, just explaining the words itself. Let's say for example, when this thing, what is that? Mendel considered this characteristic of shape of this thing, pea or seed. So this was the trait he observed and how this segregated. So that we already have in the like literature, right? So just at least that example in, let's say for example, 4-5 lines. So that much is enough, right? For these 2 laws. But this one anyway you have to explain it in detail. So that's why save the space, explain these two in brief. After that take this as separate part. Once you have taken this, so law of independent assortment. Right. So here what you have to do is that, see, first thing obviously everybody will be knowing this flow chart, right? So considering this characteristics, let's say R, and then this thing small r, y, small y. So when gametes are formed, how it will form individual gametes. So individual gametes will be formed. After that, once selfing is done, so R small 1, else Y and small y. So like this gametes will be produced after the selfing. So this is the symbol if you remember. After selfing what will be produced? Either it is R, Y. So in this way we are going to have, right? So ultimately what is the ratio we are deriving from this? What is the ratio? 9:3:1. So remember this. After this, see what exactly it is conveying, that has to be written in the words, correct? That is one. Second one is that actually when these gametes are formed, let's say for example could be R, or else could be this R, or else Y, or else this big Y. So there was 50-50 percentage for each of this to be expressed. Are you getting? In other words, under this law what it is trying to say is that there is equal opportunity for every allele to be expressed, right? So this also you need to explain it. Now the problem, or else like the mistake students make, is that they will write this flow chart and they will also give this 9:3:1, but after that they will forget about to bring this into the context of law, because according to this example how law of independent assortment is showcased, that has to be written in the words. Are you getting? At each stage what you can do is that write this in half of the page, right? Are you getting? So this is what I am trying to say, right? So make column this much space you have to be left, and in this space just write about the flow chart, and at each stage try to reiterate what exactly is the law of independent assortment according to this example. Are you getting? So this part is very critical, and this is where students will make the mistake. They won't write this part. See, without this part what I will be assuming is that okay, you are biated and you are good at representing what are the things it is there in the textbook. But if you have really understood it, explain those things in the words. Clear? Yes? Now, so for conclusion what you can do? Conclusion? Yes? See, you need to think, because for everything if I go on saying I can say, but you won't be like thinking in that way. So for this what should be the conclusion, or what could be the conclusion? Anything you can think of? See, what are the things we discussed? One is laws of heredity in general, and then law of independent assortment. Right? Now can I say what could be the conclusion? One way. See, for example, if you take any law for example in the world, can it be universal? Can it be universal? You know, universal, right? There are no exceptions. Is that true? No, right? There will be always exceptions, correct? What is the exception for law of this thing, independent assortment? See, come on, you should be thinking. See, these are the basics. Okay, even before that. Okay, when will this independent assortment will happen? Correct. So that is correct. But for this thing, law of independent assortment, in the context of cell cycle, when it will be appearing? No. Metaphase of? See, meiosis. See, why are you making it so much complicated? See, this is not to be biated. See, I said gamete formation. When will the gamete formation happen? In mitosis, will gametes come? It is in the somatic cells. I mean in the vegetative cells, correct? So this thing, meiosis, will happen in gametes, reproductive cells, correct? Just by that knowledge can't you say? In introduction, I mean in this body also you can incorporate that. See here for conclusion what you can do is that though this law of independent assortment has given the wisdom about understanding the plant kingdom or else inheritance pattern between the organisms, it is having the limitation. The linkage or else linked genes is an exception to this theory. that you can write. Or else by utilizing this independent assortment, have you achieved anything significant? Yes or no? That we can do. For example, what you are saying is this thing, hereditary diseases like, for example, hemophilia, sickle cell anemia. So all these things we can understand and we can establish the connection between the family tree. That you can do, definitely, because it is all in this gamete formation and it will be transferred because it is a characteristic which will be transferred. So it has to be through gamete. So definitely you can do that. Very good. See, that is what you have to be thinking. For example, I showed you one way of thinking is like disease elimination, or else where is the practical applicability? Are you getting? So one other way is like this thing. What is that? Exemptions. Or else, let's say, for example, if at all there is a practical limitation for that. So that also can be your conclusion. Saying that this is the limitation, we can break this barrier. That you can say. But for this is the law and that is the exception, we cannot break it. Are you getting? Right? But let's say, for example, if at all you are speaking about a methodology, and in that methodology there is a human limitation, then definitely we can breach that. So that could be brought in the conclusion. Clear? Yes? Yeah, definitely. So this is what I said. So when this came, where is that? This one. So here only that table will come. 9:3:3:1. Definitely you can put. So that is what I said. If you remember, I told you like this, right? So keep at least this much portion. In this, all those table plus your this gamete formation, the characteristics which you are taking, which is the plant, all those things we have to mention. Here you have to give this analysis. Clear? Yes? Everyone? Okay. Test cross. So for this it is not needed. Not needed. Or else in the explanation you can mention what is the result of the test cross we are getting. In the result you can mention, right? Because space is a constraint here, so putting the table, explaining everything will take time. So just like highlight that test cross ratio which you are getting and just write in one word. So that is enough, right? Any doubt? See, these are like established ones, or else content is not a problem here. So that's why I am moving little bit faster, right? Next question, fourth question, right? It is about chromosomal aberrations. Okay. Enumerate various chromosomal aberrations with their types. And explain in detail inversion type of chromosomal aberration. So this is for 15 marks. So 3 pages will be there. So take time and give me the structure. Is it done? Shall we discuss? Okay. Enumerate various types of chromosomal aberrations and explain inversion type, right? And again, this chromosomal aberration, it is one of the most repeated questions, right? There are only two categories. One is numerical and structural. So at least on one type there will be question, or else in this way, like generalistic one, right? There will be the questions. Clear? Now, see, apart from like what we are going to discuss today, for this topic, what you have to know is that apart from like one is of course the types, the concept, etc. Second one is like the applications of that. See, individual applications you may be knowing, like aneuploidy, polyploidy, etc. So what exactly it will be used. As in general, the whole idea of like what is the usage of this chromosomal aberrations as such, right, and limitations of it, right. These two things you should be known to you. The overall thing, not just like individual one. Clear? Right. Okay. Okay. Before we start this discussion, for example, you would have seen this housefly. Yes. Correct. Housefly belongs to which family? Entomology. It belongs to Diptera. Good. Correct. It belongs to Diptera family. Now, why I have taken this is that if you have like, of course, one thing is like disgusting feeling which we'll get by looking at houseflies. Apart from that, if you have like closely observed this, so there will be variations in this. What is that? Wings color. There will be variations in the color near this mouth, or else there will be a lot of like morphological variation which will be happening. Have you observed? Right? Though it belongs to the same species, why does that differences is there? One example we can, or else one assumption or else like reason we can give is mutation. Obvious one. But is that really possible within the species in the same time that much mutations will be happening? Is that possible? It is difficult, right? So this is a puzzle, correct? A evolutionary puzzle. Are you getting? And one of the reason which we can give for that is, again, it is leading to the inversion type of this chromosomal aberration. And again, it is not clearly established, but we are knowing that, okay, this could be one of the reason. Are you getting? Now, can you interlink like why this topic is important? As now, are you having the interest to learn? Right, so this is the important thing. And again, as I said, so these things were not taught to us, right? I learned it in a hard way. Because see, I actually love this nature, and I've been like roaming around in this jungles and all. So when I come across some species, so I'll be naturally curiosity, like natural curiosity will take over for me. Like if you want, I can give you like food for thought. Now let's say if you have observed this, okay, if I bang you onto this door or else on this let's say wall, so your head will be damaged, or else at least you will be feeling giddy, you will be not feeling good for some time, correct? So that will be happening. Now in nature we have one spectacular bird, that is woodpecker. And woodpecker, you know, the beak would be sharp, but still it is putting its head back and forth on the same wood again and again, but still it is not suffering any brain damage. How? So think over it. If not, just Google it down. Because these are the things you should be naturally curiosity for you, and then you think over that, then you will get certain concepts. Are you getting? For example, if you have been like following this UPSC pattern, so this is how they are asking in this environment questions. For example, this year there was question on waggle dance, yes, bees, and is like bees are doing it for just for fun? Communication, what sort of communication? Correct. See, in fact, now we know that, and scientists have found the pattern. That is, bees actually they will do this sort of waggle dance, like in this way once, and they will complete this circle, and again they will complete one circle. So how they are completing these circles, how many circles, and the pattern in which it is making, it also indicates the distance, where it is present, in which direction it is present. Everything can be communicated with this, that waggle dance. Are you getting? But to the common eyes, how does it looks? It is just dancing. Are you getting? Without any pattern. But there is a hidden truth in that. Are you getting? See, there is lot of unknown things in this universe than what we know. Right? So be humble and let's learn more and more. Right? So this is one sort of that example, because chromosomal aberration, it is sort of like black hole. Because in black hole, all that we know is nothing. Exactly. Right? So after so many years, we got to know, okay, this is how it might be looking. Right? Interstellar is one of the example where all the theories and like fascinations they have put there. but essentially how exactly it looks and traveling to the wormhole and all those things, it is still a fantasy, right? But same thing is true in the case of this chromosomal aberrations. Could it be like this thing numerical, or else could be it of structural? Are you getting? So that's why when you are writing this answer, have this fascination while reading at least. Are you getting? So while writing, obviously that will be reflected. Clear? Okay. Now can you give the structure for this? Structure? Obviously you will be saying, sir, we will be writing various types of this chromosomal aberrations, and then we will be going for explaining this inversion one, right? So that is pretty correct only. See, okay. So intro, how can we start this? Intro for chromosomal aberrations. Come on guys, you should be giving me this. How can we start? Definition of chromosomal aberrations. Okay. Okay. In this, can you make anything specific? Okay. Why do these chromosomal aberrations occur? Why? Come on, say something. Mutation. Okay. Okay. One reason is mutation. Fine. Could be spontaneous. Could be like induced. Whatever. Okay. Then. What is the basic reason? Let's consider, see this mutation in the nature, it is happening at very slow rate, right? So it is over so many generations, but still there will be chromosomal aberrations. So apart from mutation, can you think of anything else, the fundamental one? Correct? Gene sequencing, still more fundamental one? Correct. So replication is correct, but it is basically of this cell cycle. So that's why again and again I am saying, see this knowledge is very, very vital for any topic. Clear? How? See, for example, if you have been through the cycles, different stages, you know that. So this is the plate where like chromosome itself will be like, what is that? Arranged itself. Now while it is moving towards this, spindle fibers will be formed, right? So it will be dragging certain part of the chromosome. Correct? Certain will be moving there, certain will be moving down. So what will happen? This process is called as disjunction of chromosomes. While this is happening, at times what will happen? It will either like pull up lot of things, or else it won't pull up in the proper way. Right? Again, as I said, this is a dark area. We don't know exactly why it is happening, but it is happening. Right? So this is one of the reasons. I'm not saying that this is the only reason. Are you getting? So this is one of the reasons how like crossing over will be happening. So during crossing over also there will be loss of certain segments. So that actually it is called as breaking points. In this context, chromosomal aberrations, correct? So when this breaking point happens, so let's say if at all this is the chromosome, if at all breakage is happening here, so let's say this could be A, this point, and this could be B. So now after rearrangement it could be BA, or else this part itself will be eliminated. Are you getting? So all those things is happening in the cell cycle only. The fundamental understanding. Are you getting? So like for introduction, what you can do is that the abnormalities or else anomalies in the cell cycle, especially reflected in the way of non-disjunction of the chromosomes, is leading to the variations in the chromosomal numbers and structure, which is quantified as chromosomal aberrations. Are you getting? So there the keyword is non-disjunction of the chromosomes. So disjunction is important, but it is not happening at the proper way. Are you getting? So non-disjunction. So remember the word. Non-disjunction. Clear? So this is one of the reason. So you can start like that. After that you can say the classification. Again for the classification I have given the diagram in the Telegram channel itself. So here like let's say for example this is for 15 marks, right? If at all this was for 20 marks, what could have been done is that all those like things we are going to write in this diagram could have been explained at least in 2 statements. Let's say for example if you are saying this nullisomy, give the condition, right? What exactly is the nullisomy? And monosomy, give the condition. If you are saying about like other conditions, at least mention what exactly it is, if at all it is for the 20 marks. Here again the keyword is saying as enumerate. So if you remember our first class, what did I say? Enumerate means it is listing down. Are you getting? You have to just list it down. So that list has to be comprehensive, and you just list it down various types of these chromosomal aberrations. Within that, what are the subtypes we have? Clear? So this much you can do, correct? Okay. So what are the, let's say for example, numerical aberrations we have? Yes. Polyploidy, aneuploidy, correct? Then we have trisomy, monosomy, correct? For this thing, structural, deletion, duplication, then inversion and transmutation. So these are the types we have. Within this also we have subtypes, right? Just mention those things in the flowchart manner. That is good enough. Clear? Now, second part is important. See, all these things it will take hardly half page, but still you are left with almost like 2 pages, right? So 2 pages actually you have to explain this inversion in detail. But again, the challenge is, sir, all that we know is in inversion, one is pericentric, paracentric. After that, there is no knowledge, right? That is not true. We can write by utilizing that also. See, for example, inversion type of chromosomal aberration. First thing, it is structural aberration. If at all it is happening via structure, so most probably the reason could be, as I said, it is non-disjunction of the chromosomes. That is one. Or else could be mutation. Correct? So these two reasons you should be keeping in mind. And when was this, or else in which organism this inversion was discovered? Any idea? You know that it is a special type of chromosome. Now can you say? You know special types of chromosomes, right? In that one special type is there. Which is that? It is salivary chromosome, salivary giant chromosome which is there. In that this inversion was for the first time discovered. Clear? Now, so these things it will just establish this basics. As I said, like start from this basics, like from why does this inversion occurs, right? So basically inversion, as I said, one is non-disjunction of the chromosomes and mutation. So these two reasons, just explain that. While this thing chromosomal crossing over is happening, one is improper crossing over, second one is non-disjunction of the chromosomes. It will lead to the breaking point of, or else it will lead to the breakage of this chromosomes, and while rearranging there will be variations in that, so that will be giving rise to this inversion. Correct? So basically what is the meaning of this inversion? The literal meaning? Ulta. Correct? Inverted one, correct? So that is one. As I said in the beginning also, if at all this is the chromosome, and if at all this is the breaking point, and A and B, so next one could be B and A, right? Or B and C, anything could be there, but basically this is what the inversion, basic understanding, correct? After that we have this types. One is pericentric and another one is paracentric. Now tell me, in which type we have this centromere being having the breaking point? Pericentric we have centromere, pakka? Yes? Correct. So here centromere will be there and breaking point could occur in the centromere. And paracentric there will be no centromere involvement. Correct? Now, so which is the most frequent type of occurring? Paracentric? Pakka? Good. So paracentric is the most often occurring one. Most often occurring one. And this is very less. And another key feature of this pericentric is that it is suppressive. That is to say, the expression itself will not be proper, and in fact it might lead to this cell death. Right? A lot of negative things are there in this pericentric. Are you getting? So in paracentric it is the most occurred phenomena, correct? So here what is happening is that there are both positives and negatives, correct? Yes. So you explain these two types plus the characteristics, and I will just give you the headings, right? So that will be much better. So what are the things you should be incorporating? One, as I said, it is establishing the concept, where it was discovered. So what is the reasons for that? That is in the causes, correct? Third one is types. Explain each type, and in fact you can give examples also. For example, this paracentric, mostly it is occurring in plants like maize, piece like this. For example, this paracentric, it will be pericentric, it will be occurring in Drosophila, mainly in this Dipteran family, mainly in the Dipteran family. That you can give types explaining them. Fourth, detection. How can we detect this? Inversions, right? That is one. Fifth one can give uses or limitations. That you can give. Clear? Yes. Now for this, how can we conclude? Conclusion. So question was on chromosomal aberrations as such, and then it was specifically on inversions. Now, can you give this conclusion? Yes? What could be the conclusion? Hybridization programs. Okay. Yes. Okay. One is hybridization programs. Okay. In a way, yes, plant breeding, it will be helpful. Correct. Then, anything else? In the beginning only I told you. What is that? See, for example, the proper knowledge or else more understanding of this chromosomal aberrations, inversion in specific will be helpful in solving evolutionary puzzles, especially like polymorphism within a species. Example, Dipteran flies. Yes, are you getting? Right? So see, if you write this thing, it is also fine. But if you put this thing, definitely it will be adding value. Because this is not like, see, all this contents, what I am saying, it is mainly derived from this research articles. Right? As I said, see this, have you heard this, this thing, journal called as Nature? Yes. So this is one of the most read magazines from this scientific community, right? So I am taking most of the articles from this, right? And again, if you go too much deep into that, the caution, if you are willing to read that, the caution is at times it will be too technical. One. The second thing is that it will be too deep. So if you are going reading it again and again and again, at the end of the day you are just taking this point as conclusion. Then what is the cost to benefit ratio? See, here the thing is, you should be smart enough to know that, okay, if you are reading this for pleasure, just for the sake of enjoyment of the subject, or else it is for the examination. When you are doing that for the examination, be smart and take what you want. Are you getting? For me, it was enjoyment, because I read for enjoying all those things. Obviously, I am not writing the exam, so that's why I will be enjoying. But for you, it will be examination. So that's why you should be having that smartness. Are you getting? Yes? Clear now? For this also, I think I posted this thing, diagram and data in this, content in this Telegram channel. So that's why I'm moving forward. Where is that? Okay. So next is sixth question, right? Sixth, correct? Fifth. Okay, linkages. That is fifth question. Okay. Short notes on various types of linkages and their detection. Okay, this is for 15 marks. So 3 pages. So take time and give me the structure. So is it done? Okay, let's start this. Okay, question is about short notes on various types of linkages and their detection. And again, linkage is one of the most repeated topic, right? So all the types, it should be known to you, right? And also like detection and usage of linkage, both in genetical perspective and also in the perspective of breeding, how it will be useful. Clear? Okay. For this, how can we start? Or else, like, what should be the structure? What could be the structure? This is for 15 marks, yes? Yes. Okay, one is definition, then types, explaining the types, and then detection, correct? Okay, so structure is pretty simple here. But again, like, what are the things you have to write within this? I am going to say. Okay, what is linkage? What is linkage? What is linkage? So we know linkage is linkage. What is linkage? Close association of genes. Okay, then see, just now I told you, if you remember, let's go from basics. So meiosis is happening. So in meiosis, finally we are going to have gametes being formed, correct? So this gametes, what will happen? Chromosomes, it will move from this thing, parent to this offspring, correct? So gamete formation will happen. So within this gamete, when these chromosomes are moving, so basically chromosomes, we know like genes and also we know alleles, correct? Let's say this is the this thing, gene. So on this, let's say for example, 2 alleles or else 2 genes are sitting here, let's say, and these 2 are like very close friends. So what will happen? They will tend to move towards the same goal, whatever they are doing, correct? Sorry. So this tendency to stay together in hereditary transmission, it is called as linkage, right? Are you getting now? Right? So let's say for example, tallness, right? So tallness, if at all this trait or this character, it is governed by, let's say 2 genes. One actually it is like over dominant, it can be expressive, right? And another one recessive. So depending on which is coming to you, then that will be expressed. Let's say for example, if at all tall plus another character, it is linked together, right? And both will come together means both characters will be expressed. There are high possibility of that. Let's say for example, one is for tallness and another, let's say for example, it is for hair, right? So now if at all it is say tall and let's say for example good hair, you will be getting both. If not, it will be other way around. Are you getting? So this tendency to move together of 2 genes, it is called as linkage. And the genes which are having the tendency to, those are called as linked genes, right? Linked genes. And as we discussed earlier, this is the exception for independent assortment, right? Law of independent assortment. So this much you can establish in the beginning, correct? Yes. After this you can give the types. So how many types are there? How many types of linkages are there? 3 types. What are those? What are those? Never heard of linkage? Forgotten, already forgotten. See, basically we have like 3 set of classification. One is based on crossing over. So based on crossing over is what like most of the people will be knowing. One is called as incomplete crossing over. Second one is complete. So in incomplete, what is happening, or else what is the main outcome we should be knowing for? We'll be looking for. Basically, like for example, let's say if at all crossing over is happening, is it preferable or is non-preferable? Preferable or not preferable? It depends. So what is the character we are getting, correct? Basically, crossing over, if at all it is happening, what basic phenomena will be happening? It is recombination, correct? Yes. So here it is incomplete crossing over. Whether there will be recombination here? There will be no recombination. So this is the important one, right? If at all no recombination means there is no variation. If at all no variation means There is no better development of the character. Are you getting? Now complete variation, of course here there will be recombination. Clear? This is one type, classification based on crossing over. Second we have is what is it? Based on this genes, right? That is, one is called as coupling phase. Coupling phase. So here what is happening in coupling phase? Let's say if at all this is the gene. So on this, like dominant character will be on one side and recessive will be on another, right? So this is one. So second one is called as coupling one, and what is that called as? Repulsion, right? Yeah, repulsion. A repulsion type. So here what is happening? One dominant, one recessive, one dominant, one recessive will be there. Correct? This is one. And another one is based on the chromosomes where it is happening. Chromosome. One is autosome and another one is allosome or sex chromosomes. Clear? So this is the basic classification we have. If you want, you can give this in like flowchart manner in the beginning. After that, you need to explain at least each type here, right? What it is and why it is happening. Are you getting? So that much it will be taking at least like more than 1.5 pages, right? So that much it will be taking. After that, detection is there. So for detection, which are the methods we have? Correct? What is it? Correct? No. No, no idea. One is test cross, yes or no? Another one is chi-square. So in test cross, what is happening? What is happening? We are taking F1 into double recessive parent. In fact, if we look at it in like, let's say, ethical perspective, or else let's say for example from whatever the humanism we have developed from that way, so then whatever the plant breeding we are doing, so that is against to the nature, right? But for us, let's say for example, to just accept the third gender itself is taking time. We are not accepting that, but here we are doing howsoever we may be feeling. We have this so many classification, let's say for example, incest is prohibited, or else let's say for example, close relations should not be married, etc. So many things we are doing, but when it's come to plant breeding, everything is permissible. Are you getting? Just because they don't have the voice, we are doing whatever we want. Are you getting? So this is humanity, we say. So that's why these sort of like dilemmas, or else let's say arguments, if you are bringing in, one is essay, second one in your ethics answer, definitely it will be sounding much matured. Are you getting? Because you are thinking in a holistic way. Are you getting? Yes or no? Because when you are saying like, let's say, what are the differences between this ethics, morals, values? So what you will be writing? So you will be taking the classic definitions, you will be by-hearting them and writing. But if you bring this sort of subjectivity in them, so believe me, it will be a very good answer, right? So, and also in essays, right? Okay. So that is for other day. So, but essentially this is what it is. What is that? Test cross and chi-square test we have done in like UG, right? We'll be developing that table, we'll be applying the formula, and then we'll be getting the data. So these two are the processes we have for detection of the linkages. Just explain them in brief, that is more than enough. Clear? Now, again conclusion. What is the usage? Like think of like usage of linkage. What is the usage of linkage? There is no usage. No usage. If at all two like genes which is having linkage and both are like contributing in a good way, then that will be beneficial. Yes. For example, linkage as a concept, it has been explored very intensively in maize, right? So this is one, and you can say that proper gene mapping of a crop and understanding the linkage patterns will be helpful for us to manipulate them in our beneficial way, right? That can be done, right? You can say about disease resistance, you can say about pest resistance, you can say about increasing the agronomical traits, everything will be possible, right? So that you can give in the conclusion. Clear? Right? And again, see, I will just give you this classification if you want, but I think classification is known to you, right? And it is also available in the literature. So we'll move to the next question then, right? See, for all these things, The content is not the problem. How you are presenting it and how you are concluding it and how you are starting it. So that is the main intention here. Clear? So that was question number 5, right? Okay, next is question number 6. 6 is, okay, aneuploids, right? That is the question. Aneuploids are generally weak, but they are helpful in plant breeding. Explain the statement with available info. This is for 10 marks. Yes, Wahid, that is correct. Okay, is it done? Okay, we will discuss. Aneuploids are generally weak, but they are helpful in plant breeding. See, in fact, like the same statement you can find in what is it? Pundan Singh or else BD Singh, any textbook you read, or else any person who is speaking of this aneuploidy, definitely has to say this statement, right? So that's why, like, understanding that statement, why that is weak and how that is beneficial. So that is what, in other words, if I want to simplify this, how can I put this? How can I put this in simplistic words? Or else can I say, for example, advantages, disadvantages, applications and limitations? See, same thing, if at all it is conveyed in some other way, people will be scared. Are you getting? So look it at in a simpler way, right? Okay, so before going to this, so you would have heard about this term, Yes? What is it? Brexit, right? What is Brexit? What is Brexit? You would have heard, right? Yeah, what is it? Yeah, so British, that is UK, it moved out of European Union, correct? No. UK. See, one of the reasons what UK had given is what? See, one was employment, correct? Second one was resources. Third one was increased migration, climatic refuges, and increased this political refugees, correct? These reasons they had given. Now let's say hypothetical numbers I am saying. If at all the UK population is 100 people, and if at all in European Union, let's say for example there are 1000, and always there are wars between different nations. You would have heard about this Spain, within Spain also there is wars going on, yes or no? If not, you should be knowing that separatist movement is going on. Now if at all this is happening, obviously they will be moving towards this, correct? Now what they are saying is that, see, have a resources or else capacity to manage 100 people. Now if you are bringing some people to here, what is happening? For me, management is tough, correct? And let's say for example, if at all UK people move out from this little bit more, when like what is incoming is less, then that will be balanced out, but that is not happening. UK people are trying to reside in the UK, but addition is coming. In these cases there will be conflict, yes? So everybody cannot be saying as like India. So India, actually we have this something called as tolerance, unity in diversity, etc., correct? So saying all those things, at least we are being satisfied, though not to the fullest extent, at least by mandate of the constitution we are living by that, yes? Now what is the relevance of that year is that simplest thing. See, what is the meaning of Aneuploidy basically? What is the meaning of Aneuploidy? Forgotten. What will be happening in Aneuploidy? Either there will be increase, that is addition, or else deletion, or else reduction in the chromosomal number. Yes? Yes? For example, if at all there is this euploidy situation, or else let's say for example there is this ideal number, like each species should have this sort of chromosomal number. Let's say that is somewhere around 40, right? If at all that is the situation, that is to say each individual 40 genes or else 40 chromosomes that will be performing certain function. Now suddenly if I reduce this to 39, or else if I increase something to 41 or 42, now what will be happening here? Either one, the performance of that gene which was supposed to be doing, that will be repressed, that will not be happening, or else there is something else which is coming and it might be dominating over others. Are you getting? Yes? Now can you apply the same concept of Brexit? That is the fear they are having, yes or no? Same thing is going on in USA. They also say that Greater America belongs to only American people, migration should be stopped. In fact, that was one of the propaganda of Trump, yes? Are you getting now? So how this current affairs can be like interlinked to the concepts. If you understand in the way, see, everything it is happening for the resources. Yes, same thing is true in this genetics also. So 40 was the number. Now either it will increase or reduces, there will be effect on certain functions, right? So just keep those things in mind, then we'll come to that. Now can anyone give the structure for this? Yes? Structure? See, as I said, look at it in the simplistic way, that is limitations and usages, that is what it is asking, correct? So how can we start the introduction then? Obviously one form is definition, correct? So definition is the one way, and in fact it is one of the best way also, right? If not, if you want, what you can do is that you can start with the variety developed utilizing this aneuploidy condition, right? That is also one way, but here I prefer this concept based only, because the question itself is focusing on more on concept, right? So here, as I said in the beginning also, you can start like this. Either because of mutation or else non-disjunction of chromosomes, there will be variation in the number of chromosomes or else structure in the chromosomes. So when there is addition or else deletion from the basic chromosome number, then it is called as aneuploidy. Are you getting? So that much you can do, correct? After that you can give the classification, correct? classification can be provided and give the conditions here of each type, correct? This much you can do. Now, important part after all these things is, see, this would have hardly taken like for you, let's say somewhere around maybe quarter of the page. After that, what you have to do is that reasons for weakness, why it is weak, correct? Now you remember the concept which we are starting, which I told you in the beginning, that is ideal number would be there, from there there is deletion or else there is increase. Now what will happen? Basic thing, first and foremost thing, what will be happening if I lose something or else if I increase something? Let's say for example, I will put it in a much simpler way. Let's say now if you are not writing the mains and if you are attending this program, you will be happy, you will be enjoying and you will be learning. Let's say for example, the pressure of examination is also there and you are in the class and I am saying some story which is not relevant for the exam. Now how will you be feeling? Pressure will be more. Basically your behavior will be altered. So that behavior in genetic terms I can say it as gene expression. Yes, the gene expression will be altered here, yes or no? And then this gene expression, actually what will happen, either it will be over expression or else under expression. Both are not good, correct? Next one is I can say is gene imbalance. Gene imbalance, so the performance of each gene will be altered, correct? Then can I say increased or else reduced variability? If at all, see basically all this breeding methods or else breeding program, it is running behind this variation only, because I want to create variation in whatever the natural thing is happening, correct? So that will be altered here, correct? Then any other reason why it could be weak? Yes, if at all gene is altered, what other things will be altered? Very good, very good. Protein. If at all protein is altered, can I say that protein is also needed for resistance? So if at all resistance is lost, again percent disease susceptibility, it will increase. Are you getting? So considering all these things, we can say that yes, this thing aneuploids are generally weak. Are you getting? So this is one part. So for second part, we can say the advantages basically, right, or else applications, or else in actual words what you want to write is that how it will be helpful in plant breeding, right? So what are the uses of aneuploidy in plant breeding? Yes? No uses? No uses? Okay, resistant varieties. Then, see here actually what you can do is that though they have asked exclusively about plant breeding, you can broaden the scope. For example, it can be used in this, what is it, biotech. In biotech, utilizing this genes, or else, let's say for example plasmid mediated gene transfer, we can again increase the number, or else we can like rectify it, or else just by lowering it I can get something beneficial. Let's say for example, if I have done this genome mapping and I know that this gene it is coding for this diabetes, by eliminating that I can be eliminating diabetes. Are you getting? So those will be the helpful ones, right? So any other like applications we can write, and for conclusion what you can do? For conclusion, yes, we are speaking of aneuploidy, its advantages and disadvantages. What we can write as conclusion? Good, good. Down syndrome and Turner syndrome. So this can be reversed or else understood properly, correct? Then anything else? Anything else? Aneuploidy. Disease resistance breeding, right? So this also we can do, correct? Or else let's say for example drought resistance for this dryland agriculture, that also can be done. Yes? So in this way we can write this answer. Clear? Yes? Are you guys learning? Yes? Enjoying? Online people, are you enjoying this session? See, all those things I need not say. Like for example, starting from some interesting thing and then leading to the concept. Obviously, that is not what I had promised. But see, for me, these classes, as I said in the beginning only, this is just not just for this money. It is for me also, I should be enjoying, you also you should be enjoying. So ultimately, we are like, what is that, children of this science. So we are all explorers here. So that is my attitude, that's why I will be teaching all those things. Clear? Okay, so I will just give you a few things, you can take it down. Why aneuploids are generally weak? Why aneuploids are generally weak? Why aneuploids are generally weak? First one, gene imbalance, gene imbalance, gene imbalance, put an hyphen. Either it will lead to overexpression or underexpression of certain genes. Next point, essential genes missing of an essential gene may lead to functional impairment. Next, chromosomal imbalance. Number of chromatids will be altered during cell division, which interferes in spindle fiber formation and may result in cell death. Next one, developmental abnormalities. Abnormal development of embryo. Next, congenital malformation. CONGENITAL. Congenital malformations and miscarriages. Next point, reduced viability. Because of less reproductive fitness. Next, applications. One is gene mapping. Next, genetic stocks. STOCKS. Genetic stocks. Next, chromosomal engineering. Genetic stocks is basically like germplasm. We'll get to know like how many chromosomes will be present, all those things, right? Next is study of polyploidy. Next, biotechnology. Next, cancer research, oncology. Okay, next question is classify mutation explain the procedure for induced mutation breeding. This is for $15. 3 pages. So take time and give me the structure. Okay, is it done? Right, okay. See, before we go to this, I will just tell you like a way to remember this procedures, because there are so many procedures, so you have to remember, right? So my like view or else like point of view of like reading this procedures is simple. It is from cooking point of view. How many of you can cook? Okay. See, basically, let's say for example, if you want to cook this biryani, so while cooking biryani, the ingredients which you are going to put, that has to be in a specific order. If not, flavor will be altered, texture will be altered, or else rice will not properly cooked, correct? All those things will be there. So same thing is true for breeding. But have you ever like burdened yourself by like by hearting what is the procedure you have to do for cooking? It will come with practice. Same thing here also. Are you getting? Right? Let's say for example, when you are saying about this thing, any procedure in plant breeding, first thing is that initial material has to be created, correct? So could be from mutation you are going to create, or it could be from, let's say, this thing, clonal propagation, basic material you are going to create. So that basic material itself is not enough. First you have to multiply that. So in the first stage when you are multiplying, there only will be going for selection. That is not possible, correct? So that first stage is just multiplication. After that, in that you are going to select the seeds. Then in the third generation when you are going to plant, then you will be going for selection and then evaluating it whether it is up to my standards or not. Then again the selected seeds will be sown. Here what you will be doing? Within that there is a comparative analysis. Okay, this line is better or this line is better. Are you getting? Then within that the good performance, then you are going to select it. So then what you will do? Then it will be of yield trials, correct? Multi-location yield trials will be done at least for 2-3 generations, and wherever it is performing better, then we are going to release that as a variety in that particular location. Are you getting? So this is the simplest thing. The flow of things, it should be there in your brain. If you take any procedure for that matter, believe me, in this story format only it will be going. Only thing is that either here it will be mutagen. There it could be, let's say for example, taking 2 characteristics. Could be of disease resistant from one ruling variety, one could be from land race, and then combining together, then first initial material will be produced. Are you getting? The way to produce that initial material could be different. So that is where essentially we have different types of like procedures. But within that selection again, like for example mass selection, we are just going to select it in a random way, like whatever it is there we are going to take it. But let's say for example pure line, within that which are the pure ones, that is like in selection procedures we have different thing, but essentially that flow of chart is same. Are you getting? So understand this philosophy, like how we have to remember, then rest of the things will be easy. If not, you will be end up like biating, okay, this is M1, this is M2, or else this is F1, F2, where I should be writing F2 to F10, where I should be writing F2, F3. So those biating nature is not at all needed here. Are you getting? Right? For us also it was happening the same. See, like I am not criticizing the faculty as such, like during our UG. What used to happen is that, see, that might be his teaching style, I don't know. But the thing is, like you would have written something in the paper, you would have come, even before explaining something to us, you would have write the same thing, whatever it is there in the paper on the board, then you will explain the same thing, whatever it is there. So essentially you are not at all getting like, okay, what is this, why this is important, etc., etc. So for that again we have to put the brain, then we have to understand it, so that is how it happened. But for you it should not be the case. Are you getting? You should be enjoying it. Are you getting? Basically, see, breeding is one of the easiest thing you can learn. Clear? So as I said, see, mutation, it is part of genetics, but the procedure for mutation breeding, it is part of plant breeding. So this is how they are going to combine the things. Clear? No. So how can we start this answer? Yes? How can we start? Definition. Definition of mutation. Correct. Then? Okay. Varieties. Using mutation breeding. Correct. Then? Can we say the need? Yes or no? See, because if you take any other breeding procedure, what is happening? You need lot of time to create the variation itself. Here already variation is created, then you are going for multiplication process. Are you getting? Yes. So this is one way, or else GMO, genetically modified organisms, where we can get initial variability, then we can multiply. Are you getting? So time taken to produce itself, it itself will be less here. Correct? So though it may take somewhere around 12-13 years, so that is good enough because we already have the variability. Because in traditional breeding methods, what will happen, to create the variability itself, it will take a lot of time. Are you getting? That is correct. That is correct. That is why when you are doing this, what is that, mutation, so initially only we will be knowing. Let's say for example, you have certain mutagens. This thing, X-ray is there, gamma rays is there, and also like certain chemicals are there. By utilizing that, we know that if I use this in this concentration, so this might be coming. See, at the end of the day, it is always luck factor will be there. So for example, when this James Webb telescope was launched, same thing, like the leading scientist himself was quoted like this. For example, if you achieve anything significant in your life, could be in science, could be in any walk of your life, and if you are just saying that it is only because of your potential, then you are a liar. Because there is always a like factor of luck. Because even, see, there was something called as this single point failures, right? Single point failures. And this mission, actually it had more than 300 single point failures. What is the meaning of this single point failure? It is simple. For example, let's say if all the body, etc., of it is there, if at all one single point failure is occurring, means entire mission is waste. It is going into the dump. Are you getting? So that sort of thing was more than 300. Now imagine how many odds were there, right? Because it could be in any phase. See, for example, launch, after reaching the orbit, after reaching etc., so many would be there, right? Even after, let's say, all this 300 is also done, if at all some X thing happens because of, let's say, sudden solar storm or something of that sort happens, then again it will be failure. But still it is running, means there is X factor. So same thing is true for mutation also, or else any plant breeding for that matter. Clear? So that is there, but see, considering other methods, so this is much better. That is what we can say, correct? So this also you can use, the time consumption it will be less, and the product which we will be getting, it will be much better. So this is like the ways in which we can start, correct? So what is the definition of mutation? What is the definition? Sudden, correct, that is important. Sudden heritable change. So this is the important word, heritable. Because if we are creating a change and it is not passing to the next generation, it is not at all a useful thing. Are you getting? So it should be inheritable, right? So that is the most important word you should be using, right? It could be sudden inheritable change. Okay. Now this is done. Now classification. See, for classification there are so many ways, and I have given in the Telegram channel also. Based on a mutagen, again we have. Based on the site where you are going for mutation, again we have. So like I have given, I think more than 7-8 types in the Telegram. So you can go through that, and just for each one, in the bracket only I have given note. Note means how we are starting that procedure. So you can give that. Clear? So for procedure it is again simple, as I said in the beginning only. So it starts from 0 generation. 0 generation because we are creating the mutation. Create? We are creating the mutation. So here it could be like X-rays, gamma rays, or else anything. You create this mutation, correct? So once that is created, we are calling it as mutant, correct? Now in M1 generation, what we are doing is that whatever the like mutants we got, so here only idea is for multiplying. So no selection. Here there is no selection in M1, correct? After that, in M2, what we'll be doing is that here we'll be growing this the crop. From here we will be going for selection and evaluation, right? From next is M3 to, let's say, M5. For all these generations, what we'll be doing is that in this the better mutant lines will be cultivated. Correct? Again from 6th to 8th generation. So within this comparative analysis will be done. So whichever is performing better, so that I am going to take, and then from let's say 9th generation till 12th, I will be doing this MLTs, that is multi-location trials. So those which will be performing much better performing lines, it will be released as varieties. Clear? So this is the general flow of things we can represent, and give this flow chart also. And if at all like space is less, then what you can do is that they concise this in one half like this. In this one half, give this flow chart, and in this half, you give explanation. If at all space is there, then give this flow chart and then explain this. Are you getting? Right? Clear? So for conclusion, what can be done? Conclusion. How it can be used for? Food security, okay. Food security is one aspect. As if you want to speak from like breeding point of view, you can definitely focus on time. Because, so you can say like this, entire breeding programs are dependent on creation of variability. Since we are starting here, since we are starting with variation here, so it will reduce the time in development of the varieties. So that also you can do. Clear? Yes, okay. Then we'll take the last question. It is self-incompatibility. Explain mechanism of self-incompatibility. Describe its relevance in plant breeding. And this is for 20 marks. So 4 pages would be there, or else 3 pages would be there. Okay, is it done? See, before we discuss this, I will just let you know this. See, for example, if you want to understand the self-incompatibility, the first and foremost basic knowledge you should be having is floral biology, right? Considering that most of you would be taking this botany and agri for your forest service optionals. So this floral botany plays very important role, right? See, for example, you know that the male part and the female part of this flowers, so that much you should be knowing, which is like stamen, style, stigma, pollen tube. So this basic structure you should be knowing to understand this, what is that, self-incompatibility, right? Okay. So this is again pretty simple. You have to explain the mechanism and then you have to like explain the applications in plant breeding, right? Okay. So what is self-incompatibility? Inability of, okay, inability of pollen to fertilize with ovule. Okay. So this is correct, but there is a major mistake, and believe me, if you write the same statement, it will be marked as wrong. Why? What is the thing it is missing here? That is also fine. Then a very, very important keyword is missing. No, it is functional. This is the keyword, very, very important. Most students fail here, right? The pollen will be functional, it will be healthy, but still there will be self-incompatibility. There won't be proper mixing, there won't be proper fertilization. Are you getting? So if you are missing this, definitely the answer itself will be wrong. Are you getting? So this is the keyword you should not be missing. It is though functional pollen will be there, there will be no fertilization, right? So that is the self-incompatibility, right? Now mechanism is asked. So mechanism, what is the mechanism we have? Yes. Okay. See, basically when I ask this, most of the students say only one thing, that is homomorphic and heteromorphic. But essentially those are types. Those are types of self-incompatibility rather than the mechanism. So mechanism means you have to take this biology into consideration, then you have to explain. So based on that, we have actually three mechanisms. One is like stigma and this pollen, interaction. Then pollen tube, then style interaction, then pollen tube plus ovule interaction. Are you getting? So these are basically like phases in which this self-incompatibility can also occur. Let's say for example when there is functional pollen and it is going and sitting on the stigma. So stigma should be receptive, and if you have observed any flower very closely, will have this stickiness. Small hairs will be there on the stigma usually. That is to like make sure that the pollen will be functional and it will be present on that stigma for some time. Are you getting? So if at all those, one is morphological modifications, second one is chemical factors. Considering these two things, we have to explain this stigma pollen interaction. Are you getting? So why that pollen, even though it sits on the stigma, it will not be fertilizing. Clear? That is one. Next one is, let's say for example, when to understand this better, for example, you know this maize, right? So silking you would have heard, correct? So all those things to happen, what should be there? Let's say for example, this is the stigma we have and pollen is sitting here. So once it is sitting, next thing is, you should be remembering that double fertilization concept, Strasburger, etc., those things should be clear. Now once it is sitting means it has to develop this pollen tube, because ovule would be somewhere here, it has to come here and it has to fertilize, correct? So this pollen tube plus this style which will be there, so it will be interacting and there will be incompatibility. Are you getting? Again, as I said, there are morphological and chemical ones, the reasons. Are you getting? So same thing is true for ovule. Let's say somehow it makes this journey until here, but in ovule again there will be morphological and chemical factors which will be inhibiting this functionality, that is basically for fertilization. It will not be happening, right? So this you have to explain in the mechanism rather than homomorphic and heteromorphic. If you want to mention about that, what you can do is, since this is a 20 markers, definitely you can explain that, saying that types of self-incompatibility, correct? After that explain the mechanism, and then obviously it is about relevance. So for relevance I am going to dictate, don't worry. So for conclusion, what you can do for conclusion here? Conclusion, it is about self-incompatibility and applications. The question is about this thing. Yes? Yes? See, basically if at all self-incompatibility is happening, so what is the disadvantage we have? Breeding, or else which sort of breeding? That is important. Basically self-pollination will be inhibited, correct? So you can say that the creation of variability in the self-pollination crops, it will be tough. That is one, correct? And let's say for example, if at all this is the case, and say I thought of something, you said something, I forgot that. Okay, self-incompatibility. Okay, selfing is, see that in other words it will be promoting this cross-pollination, or else it will be reducing this inbreeding depression. So reduction of inbreeding depression, it is a good thing. Yes, so that you can utilize in a conclusion, correct? Or else let's say for example, if we know example of variety, incidence of SI will be high in certain plants. Take one example and mention that. Clear? This is also subject I should be going, at least that. See, because for example, for agronomy and all, we took lot more time because there I wanted to give you value addition, etc., etc. But here, like since you know the content, but still I gave value addition, but still we are able to complete it much earlier. Are you getting why? Why? It is about fundamentals. Are you getting? Again and again I am telling you fundamentals is the king here. Not just in optional, it is also true in preparation. If you are really thorough with the fundamentals, no matter how much tough is the paper, you can definitely get very good marks. Clear? So these all things I am saying with experience, but the problem is, like especially students, they will take these words very lightly. Unless and until they go to this examination, they will experience by themselves for 1 or 2 attempts, they cannot realize this, right? But that would have been taken you like at least like 3-4 years of valuable life. So that much luxury you don't have. Are you getting? So main intention or else like main use of our faculties is that we can shorten your length of your journey. Are you getting? So use in that way, and like whatever the things I said from this perspective, so think over that and then implement that. So implementation is very important, right? So knowledge you can gain, but if you are not implementing, it is not going to work. Clear? Yes? So this is about genetics. Clear? Any doubt here? No? Okay. Just a gentle reminder. So like those who are not paid this fees, like half of the fees, so for them again the reminder is just complete that payment, because see, this portal is automated. You will lose the access to the portal. That is one. Second thing is that you might have known this. So we are also coming up with this classroom program. So you would have seen, I guess, right? So this is for almost 3.5 to 4 months program will be there. So where we'll also get access to this I-ARI program plus YLM test series. So YLM it is for 6.5 months, right? So that will also be free of cost. Plus this I-ARI program, it will also be available for you. Plus in the classes we are going to go, give you this, obviously the concept clarity I am going to give. Apart from that, notes also I am going to give, plus this thing, value added material. So all these things are included in the program. Yeah, so plus additional 5% discount is there. So 5% discount is there till I guess August 12th. So like if you are interested, definitely you can enroll in that. So let's say for example, the question would be, sir, we have already paid fee for this I-ARI, then what will be happening? So whatever the fees you have paid for I-ARI, that will be deducted from the cost of this program, and rest of the fees only you have to pay, right? So same thing is true for YLM. Clear? Okay. So that will be all. So thank you. So tomorrow we will meet. Sorry? Okay, sorry. Relevance of self-incompatibility, right? Where did it go? Okay, first point. Enhancing genetic diversity. Enhancing genetic diversity. Cross-pollination, within this cross-pollination, resulting from self-incompatibility, increases the genetic diversity of plant population. Increases genetic diversity of plant population. Next, preventing inbreeding depression. Preventing inbreeding depression. Preventing inbreeding depression. Next, hybrid seed production. Hybrid seed production. Next, creating F1 hybrids. Creating F1 hybrids. Next, promoting outcrossing. Promoting outcrossing. Next, conservation of germplasm. Conservation of germplasm. Put an hyphen. Conservation of germplasm. Put an hyphen. In wild and endangered plant species. In wild and endangered plant species. In wild and endangered plant species. Self-incompatibility help in maintaining. Self-incompatibility help in maintaining genetic diversity. genetic diversity within small populations. Within small populations. Next, seed production management. Seed production management. Self-incompatibility is essential to prevent unwanted self-pollination. To prevent unwanted self-pollination and maintain seed purity. Seed purity. That is it. Right? Okay. Thank you all. We'll meet tomorrow.